So the winner of this award is Bruno Godinho. And Bruno received his PhD in 2014 at the University College of Cork. He focused most of his career on siRNA delivery, specifically for neurodegenerative disorders. He obtained his PhD quite recently, but already has quite an impressive track record, including a silver medal from the Polytechnic Institute in Lisbon. Currently is working in Anastasia Korova's lab, where he's doing great work, about which you will hear more presently. Congratulations, Bruno, the floor is yours.
You all hear me? All right. So thank you very much, Annemike, for such a kind introduction, really. It is an honor and a privilege to be here at the OTS today presenting data from the Korova lab, actually, that the Korova lab generated for the last 5 years. I generally like to start my presentation showing this figure that Anastasia and Jonathan Watts published in late 2016 that shows actually the enthusiasm of the field towards RNAi technologies. And you can see the hype, the initial hype, the first clinical trials, and then the first clinical trials failing, and then the dark ages of RNAi, and then some renewed interest in the technology. And if you actually look at the clinical landscape, so the clinical trials per year, the curve that they came up with from the top of their heads really mimics the number of clinical trials per year. You see here the enthusiasm with the first, the drop after the failure, and then some renewed interest and some maturation of the field. And actually, the maturation of the field actually came in the form of a greater number of phase 3 trials, but not only that, but also as the approval of patisiran in August 2018, which they actually predicted very well too. What I also like about this figure is that they talk a little bit about the evolution of the field, moving from minimally modified, unformulated siRNAs to formulation approaches, and then later on to more conjugate-mediated approaches. And you can see that on this figure too. Initially, most of the approaches used unformulated siRNA, minimally modified. Then you can see that the formulation approaches take over for a while. There's some failures. And recently, we use mostly very stabilized oligos using conjugates to deliver them to specific cell types. And the field is now dominated by GalNAc conjugates, and the clinical landscape also shows that. Most of the clinical trials and drugs in development are actually for hepatic diseases, and you don't see anything really for CNS disorders. No drug, no clinical trial for RNAi. Okay? And although there is a lot of money being poured into the field of CNS, there is yet to be disclosed a structure that promotes, broad distribution and also effective knockdown, sustained knockdown for long periods of time. And it's about exactly this that I want to be talking about today. I want to talk about the Dyess that we came up with, and probably you listened, Anastasia already talked about the fact that on siRNAs you can optimize the Dyess or the chemical architecture and the conjugate to which determine the biodistribution and retention within the tissue, somewhat independently to what the pharmacophore is. Okay, so you can then plug a different sequence and use that scaffold to deliver to the tissue of interest. So we started very simple. And here is a model molecule. It's a fully modified asymmetric siRNA. It has a cholesterol conjugate at the 3' end. And we've shown in cells and also in animals later on that these compounds are able to load very well into the RISC complex as good as unmodified siRNAs, giving somewhat the same type of potency. Only the compounds that were conjugated to a lipid moiety have self-delivering capabilities, and this is also true when you move into in vivo. So here we did intrastriatal injections into the mouse brain. You can see that an unconjugated oligonucleotide is poorly retained in the brain, whereas a cholesterol conjugated oligonucleotide is very highly retained. But the lipid moiety is not the only important part, and this is just a reminder. Here we did a study where we actually injected a partially modified siRNA into the brain, which was cholesterol conjugated as well. And you can see here that although we're able to achieve potent knockdown, this knockdown is short-lived compared to the fully modified counterparts. So we decided to go on this quest to, in the context of fully modified siRNAs, we decided to go on this quest to improve the delivery that we actually see from cholesterol conjugates. So here you see that cholesterol are very highly retained, but they have very poor distribution from the site of injection. So we did screen a lot of different, we generated a panel of different conjugates and chemical architectures that we looked at, and we decided to evaluate which ones work the best through ICV injections. And this ICV injection is basically an injection into the CSF space. And you can see here from this figure that the cholesterol conjugate is mostly retained at the site of injection, does not distribute very well from the ventricles, whereas other conjugates, which are a little bit less, actually significantly less lipophilic, distribute a lot better from the site of injection. And it was in this process that we were able to identify what we call the Dyess Rna scaffold. So what are the key features of the Dyess Rna? The Dyess Rna is a divalent siRNA, so it's two siRNA units linked to each other by a linker at the 3' end of the sense strand. The scaffold is fully modified at the ribose, at the whole, all riboses are modified. And we also have some phosphorothioate modifications at the 3' end and 5' end of the construct. The construct also has an hydrophobic moiety that is tagged at the 5' end of the sense strand. And we have used this scaffold with different pharmacophores, and today I'm going to focus on two of them. We have tried many others, but today I'm going to focus on the Huntington targeting pharmacophore and also on the ApoE one. So we initiated by doing a series of ICV injections of our compound into the mouse brain, and we sacked the animals at different time points and collected different regions of the brain and assess the amount of siRNA that is still accumulated within those regions. You can see that the different regions of the brain retain the oligo to different extents, and the compound is cleared over time. What is even more interestingly is that in this specific context, the siRNA concentration in the tissue correlates very well with the knockdown at the mRNA and also at the protein level. Actually, at the protein level, at 1 month, you see that we almost completely wiped out the HTT protein, so we cleared the protein to the level to the detection limit of the assay that we were using. And an NTC control does not affect the expression of the protein. Over time, you can see a recovery of the protein expression, but what I want you to focus on is on the fact that certain regions of the brain still have potent knockdown at 6 months, both at protein and at mRNA level, and those regions are the ones that actually accumulate the most initially, and then maintain that over 6 months. So really excited about this data. We decided to look more closely into the relationships between the accumulation and the mRNA and the protein silencing, and we were able to calculate the effective dose, in this case the ED50, for mRNA silencing, which was about 1.5 micrograms per gram, and at the protein side was at 0.4 micrograms per gram. The results did hold true also for a second pharmacophore, as I was initially saying. Here you see an ApoE target. We were able to knock down the mRNA almost completely after 3 weeks of injection, after 3 weeks after a single injection, and also at the protein level. Okay? And for these studies, we use half of the dose that we use for the HTT studies. So the next big question is, okay, we've done all these studies in rodents, but rodents have teeny-whiny brains. How will this translate into more complex mammalian brains? So brains that have neocortexes very well developed. And we landed on two models, the Dorset sheep and the cynomolgus macaques. And I will start by telling you a little bit about the distribution studies that we carried out in sheep. And in sheep, we decided to look at the different routes of administration that are commonly used for administration of therapeutic oligonucleotides, but also other gene therapies. So we did intrastriatal injections. We also did intrathecal injections, which are very commonly used for administration of ASOs. But we decided also to keep our standard ICV injections because we've done all that work in the rodent models. And we went a step further to implant a reservoir that permitted us to do repetitive administrations on the same animal. And I don't have time to go over all the details, what are the advantages and disadvantages of these methods, but we can talk a little bit offline if you wish later. Okay, so here's the design of the study. We injected some sheep directly into the striatum. Some other, we thread a catheter from the lumbar space up to the base of the brain, to the cisterna magna. Some other animals were injected directly into the ventricle. And finally, we implanted a reservoir that allowed multiple administrations into the lateral ventricle. So apart from the intrastriatal study, we were really hoping to see, we were really expecting similar results in terms of distribution with the CSF delivery methods. And we hoped that with the reservoir, we would enhance permeation into deep brain structures. So here's how the sheep brain looks after an ICV injection. So an injection of the Dyess Rna compounds directly into the CSF space. These brains have been perfused with PBS, so there's no interference, or we're trying to minimize the amount of blood there. So we took slices from this, and we laid them down from the front of the brain to the back of the brain. You can see here, as for reference, the intrastriatal injection gives you very localized delivery, very little delivery to cortical structures, but all the CSF methods enable broad distribution over cortical regions. Okay? The ICV and the reservoir method allowed better permeation to the striatum, as you can see here on the figure. So that we don't rely just on pictures taken from an iPhone, we did some quantification, and this is what you have here. This is of PNA hybridization assay. And again, in the cortex, you can see that the three methods of CSF delivery achieved more or less the same amount of delivery in the cortex, but the ICV method and the reservoir method were superior for delivery to the caudate and putamen. If you take a slice of the cortex and you put that under the microscope, you can appreciate that the Dyess Rna diffuses or penetrates very well in all cortical layers, and has a very distinctive localization, which is perinuclearly, and we also find the compound on the dendritic processes of neurons. Looking at the spinal cord, when we necropsy the animals, the spinal cord is very pink too. And at the bottom, you have some quantification data that shows that we have very comparable amounts of oligonucleotide being delivered through the different CSF methods. The only one that did not achieve great distribution, not surprisingly, was the intrastriatal injection. If you take a transversal cut of the spinal cord, you can appreciate that the compound actually penetrated throughout the whole tissue, both gray matter but also white matter. And if we take a picture at the ventral horn, you can see that the compound is colocalized with the neurons in that region of the spinal cord. In this case, they're lower motor neurons, important in some neurodegenerative diseases such as ALS. So we've done all these studies in sheep, and we wanted to validate if the same approach would work in monkeys. And very quickly, I'm not going to bother you again with more distribution studies. We did an ICV injection, and again, we obtained this broad distribution over cortical structures and even in deep brain structures such as the caudate and the hippocampus. We then perform an efficacy study. Here we injected 4 macaques, and had 4 macaques as controls. You can appreciate also that we have potent silencing in different structures of the brain. The more staggering results were observed at the protein level, and these results correlate very well with the amount of oligo that accumulates in those regions. Again, so just as an example, you have cortex over here, and you can see that the degree of silencing in the ipsilateral and contralateral regions, both for mRNA and protein, are very potent. So finally, we looked at the spinal cord, and we have very distant accumulation of Dyess RNAs in all segments of the spinal cord, and therefore we also found very potent knockdown of the HTT target there too. Now, really finally, we looked at this a little bit into the safety. Every animal from the efficacy study had a pre-op and a pre-necropsy MRI. We did not see any morphological changes, no brain edema, nothing like that. We looked into neuroinflammatory markers such as GFAP and IBA1, and we did not see any overexpression of those markers. We looked at some systemic markers of toxicity, and here you have liver enzymes and some CBC counts, which did not differ significantly from the pre-op values, suggesting that Dyess RNAs are relatively safe. So hopefully I have convinced you that this compound is an optimal scaffold for RNAi interventions in the CNS, and that they achieve broad distribution throughout the brain, and they're effectively internalized by neuronal and glial populations to give sustained gene silencing over a period of at least 6 months. I also show that the scaffold is amenable to different pharmacophores and has a favorable toxicity profile in the CNS. In the NHP, sorry. Part of this work has been published in Nature Biotech, and we were lucky enough to be cover of the magazine in the August issue. So this work was only really possible because we're very united group. The Corova Lab was fantastic, and doing all this work was not done just by me, so I think the team this prize is really for the team, not just for me. So I want to highlight 3 people, Chantal, Julia, and Matt. They contributed as much as I did for this project. Chantal has a poster, and Sam also has a poster, and Julia will be giving a talk this afternoon that you should definitely attend. I also want to thank the Ronin Lab and the Diphilia Lab and all the team that worked with us in the large animals. And I want to thank you for your attention. Thank you, back.
Thank you very much. There's time for questions for Bruno.
Bruno, sorry if I missed this one, but what's the specific benefit of the dye versus the mono siRNA?
Well, we believe that the Dyess RNA has a different rate of clearance than the mono siRNA. Okay, we are still investigating the mechanisms of this, but we see a very big difference between the mono siRNA. I actually don't have here, but we can converse after. We have done experiments where we injected the mono siRNA and the divalent siRNA side by side for comparison, and the clearance of the mono is a lot more quicker than the di. And there is potency difference, yes, too.
In one, sorry, Bruno?
Oh, sorry.
In one of your first slides in the mouse, you show that the siRNA, the bivalent, is accumulating, that targets the Huntington is accumulating in different brain regions. Mm-hmm. But your non-targeting is actually apparently not present. Is that not retained? Or
no, it is retained. So, so that figure, the top, you're talking about that top figure. So that top figure is a PNA hybridization assay, and we have to use a probe that detects specifically the HTT sequence. So that's just to show you that the probe is not detecting my NTC. So, but it is retained. Yes, it is retained. Yeah.
Right. So what's the motivation to use ICV injection in monkeys versus intrathecal?
That's an excellent question. So, and we also, we debated that a lot within the group. We're aware that Ionis uses intrathecal, and intrathecal is...
And everyone else.
Excuse me?
Ionis and everyone else.
Correct.
Yes.
You're completely right. So in terms of the clinical approach, IT is a more relevant approach. So, but due to the models that we picked for our initial distribution studies, especially the sheep model, we had a lot of issues with off-the-needle IT injections. And that's was one of the, so we tried to do these injections off the needle, so just basically threading the catheter a little bit, and we had a lot of backflow of the compound due to the fact that the intrathecal space in the sheep is very small.
I see.
So that counted into the decision of,
Yeah, my specific question was towards monkeys.
We have not tried IT in monkeys. I believe that
see.
an IT injection in monkey will probably be successful.
Yeah.
I know that Ionis have done that, and they're successful doing that.
Can I ask one more question? So I think the slides went through pretty quickly, but
Yeah,
if
I'm
I
sorry.
noticed in sheep, the exposure you had was all the way from 100 to 150 microgram per gram, versus in monkey it was 10-fold low. So I'm just wondering, what were the doses again administered in these two?
So in the sheep, we injected 50 milligrams, and in the monkey we injected 25 milligrams. The big difference
So it's
between the
twofold.
values, the big difference between the values is that on sheep, these distribution studies were 48 hours. The monkey values were acquired a month later.
That makes sense. All right,
Okay.
thank you.
No problem.
Last question.
Thanks for the great talk and congratulations with the
Thank
award. Just coming back to your first answer around if you take your best siRNA with your best hydrophobic conjugate and then you compare it to this very nice approach, what would you think the benefit is if you have to give a number? Like, is it a factor 2 better in potency? Is it can you kind of elaborate a little bit on that?
You're asking if I injected the cholesterol?
No, just if you have to compare this dimer siRNA with your best normal siRNA with a nice conjugate, do you then get a twofold increase in potency, or do you get longer duration of action? Just to give some numbers.
I will not be able to precise numbers, but I can tell you that it's very significant, the difference that we see between a mono siRNA and a Dyess Rna in terms of knockdown, depending on the chemistry that you use, of course. But we see very, very different knockdown. I mean, even in terms knockdown, both knockdown and the duration of effect.
Yeah.
So,
Thanks a lot.
yeah.
Okay, so while I'll put this down, you can put your hopper down.
Oh, thank you. Gotcha. Thank you. Thank you. Thank you. That's it.